Okay, we continue our study of transport across cell membranes by looking at ion channels, and then we look at active transport. Remember, we looked at, we ended the last segment by looking at facilitated diffusion by transporters like the GLUT1. Now we are going to look at ion channels. The lesson objectives for this segment: make sure that you can define carrier proteins and state the different types. Then explain the structure of ion channels and their mode of operation. Then define active transport. Make sure that you can state the requirements for active transport, and also be able to name the different types of protein pumps and state their main features and explain their mode of operation. Make sure that you can define AQPs and state their functions in cells. Channel proteins. Channel proteins. Their main features. These are proteins that are designed to have a hydrophilic transmembrane channel. If we can use this one as an example here, just excuse me. So the protein is designed in that manner, standing in the membrane, and you have that opening, which is a hydrophilic opening running across the cell membrane. The types of channels then: number one, ion channels; number two, porins; and finally, aquaporins. Looking at ion channels, their structural organization. Essentially, these are hydrophilic channels with a central constriction that allows for selectivity. Their main function is selective and rapid movement of ions like sodium, potassium, calcium, and chloride ions. The rate of transport up to 1 million ions per second. The gating mechanisms of ion channels. Defining a gating mechanism, or ion gating, a mechanism to control the passage of molecules through the ion channel. Now, before we can continue, excuse me, maybe we should kind of put a diagram here, see how we think about the design of an ion channel. Here is the membrane, and here is the channel. We talked about a central constriction. There is a central constriction. This is the protein itself. And don't forget that this is within the membrane, so here is the phospholipid bilayer. Now, this central constriction here is essentially for selectivity, to ensure that you don't have solute beyond or above certain size going through. Now, around the constriction here, you have a channel gate. The channel gate can either close the channel or move back to open the channel. The other structure that is associated with ion channels is the channel inactivating particle, which takes the form of some kind of ball and chain. This ball can come and lodge itself right here, essentially inactivating the channel. Now, going back to the gates, we name the channel gates according to the signal that operates them. If the channel gate is operated by some kind of electric voltage, then we are going to call the whole channel a voltage-gated ion channel. Some channels are operated by binding of a ligand, in which case then we are going to call the whole channel a ligand-gated ion channel. Some channels are operated by mechanical stimulation. So those are going to be called mechanosensitive channels. So let's move on and see a few more details about ion channels. This is the way we think about these structures. The only thing that is missing here is probably the channel gate. But anyway, this is supposed to show a voltage-gated ion channel. You have the channel either open or closed. But again, like I said, this arrangement here leaves something not properly set up with clarity. You have the gate as being separate from the ball and chain mechanism, which is the channel inactivating particle. A channel can be open, in which case ions can move through. If the channel gate is closed, ions cannot move through. A channel can be inactivated. That is the situation where you have the ball and chain mechanism lodged right there. This channel then is inactivated, which means it doesn't matter whether the gate is open or closed, no ions will go through. You can liken this to the door to your classroom. That is the equivalent of a channel gate. If the door is closed, you cannot enter the classroom. If the door is open, you can enter the classroom. Now, a building has the main doors which open to the outside. When those doors are locked, then essentially the building is inactivated. It doesn't matter whether the door to your classroom is open or closed, you cannot access your classroom. So an inactivated channel is similar to a building where the outer main gates are locked. It doesn't matter whether the room doors are open or closed, you cannot access the rooms. So an inactivated channel is unresponsive. It's not active. Maybe you're wondering why spend so much time studying about ion channels. Well, they are related to health and disease conditions. Here we look at some diseases that involve ion channel function or ion channel malfunction. Dry mouth syndrome, cystic fibrosis, hypertension. If you have a relative with hypertension, well, part of the problem is some malfunction of some ion channels. Diarrhea, chloride ion channels, cancer, incontinency, multiple sclerosis, and you can see a few others, including migraines, which are very common, deafness, kidney stones, and even epilepsy. So you have now reasons to study and understand ion channels and their functions. We are going to shift gears and look at proteins. These are transmembrane proteins organized as pores that are larger than ion channels. The proteins are organized as beta-glycans, and then they are lined by polar side chains. So the interior of these channels is polar. In terms of occurrence, they are very common in bacterial membranes. They're also common in mitochondrial membranes and also found in membranes of chloroplasts. The functions then of proteins to allow rapid passage of hydrophilic solids across the membranes. Aquaporins. Aqua is water, so aquaporins are essentially special porins that allow for rapid movement of water across membranes. In terms of structural organization, these exist as transmembrane proteins with 6 transmembrane segments. That means they thread back and forth across the membrane 6 times. The central pore is smaller than the pore for both ion channels and porins. We find these in proximal kidney tubules, erythrocytes, and tonoplasts. Proximal kidney tubules are found in the nephrons. So let's look at the organization of a nephron and where you have these and how they function. Your nephron is composed of a structure known as Bowman's capsule, and you have an efferent vessel coming in, which is wider than the efferent vessel that leaves. The vessel here breaks into thin capillaries, forming a structure that is called glomerulus. This is an arrangement to filter your blood. The filtered blood then comes out through a tube, and this tube comes down to form an area that is curved like this. This is what is called proximal kidney tubule. This leads to a loop which is called the loop of Henle. And then you have another convoluted area here which is called distal convoluted tubule. This then leads the urine to the urinal bladder. So where do we have the aquaporins and why are they important? The aquaporins are located in these areas here. They are very important because they allow any water that is filtered through to be taken quickly back out into the blood so it's not lost with the urine, which would cause severe dehydration. Tonoplasts, these are members of the plant central vacuoles. They are rich in tonoplasts. That's why when plants are wilted and they're watered, they can recover very quickly as they are able to take in water very quickly through the tonoplasts and fill the central vacuoles and reestablish cell turgor. Functions then of aquaporins: facilitate rapid flux of water molecules in and out of cells. The actual rate of flux here is up to several billion water molecules per second. We are now moving to active transport. All the transport mechanisms we have covered so far did not require any expenditure of cellular energy. So they were all passive transport processes. Let's look at active transport then. Defining active transport: essentially a transport mechanism that uses metabolic energy to move molecules across membranes against their concentration gradient. The requirements for active transport: you need ATP-powered pumps or protein pumps, and then you also need a supply of energy. Types of active transport: direct active transport and indirect active transport. Here we are looking at a comparison between direct active transport and indirect active transport. You can see with direct active transport, the process relies on direct hydrolysis of ATP. In this particular arrangement here, your hydrogen ions are being moved up to the concentration gradient and accumulated on the outside of the cell membrane here. The cell membrane is not permeable to hydrogen ions. The only way the hydrogen ions can move back into the cell is through this other protein here. But the arrangement is such that as your hydrogen ions flow down the concentration gradient from high concentration to low concentration, they facilitate the movement of a second solute against its concentration gradient. So in this arrangement, the second solute is said to be transported by indirect active transport. In our next segment, we are going to look at the different types of protein pumps, their design, and the way they work. Thank you.